today, the last two speakers of our conference. Everything has an end, unfortunately. My pleasure to introduce Dr. Didier ROGNAN, still fighting with PowerPoint. He is a medicinal chemist by training. He had a PhD in medicinal chemistry with the famous Camille Vermouth here in Strasbourg. Then he postdoced in Germany and at the ATH in Switzerland. And now he's research director of the CNRS, leading the very innovative in silico and also wet lab drug discovery team at the Faculty of Pharmacy. And while students, if you didn't read papers by Didier ROGNAN yet, you have a problem. You should sit down and do it. So Didier, please.
Thank you, Dragos, for a very kind introduction. Trying to keep you entertaining until the very end of this meeting. So I'm going to go back to ultra-large compound library. So you have a great talk from Matthias Rarey and also an overview by Marcus Gasreich from Biosolvity. I try to convince you why is it important now to screen very large. It's not to beat any crazy record, but because it's really useful. So I did not to convince you that chemical space is growing very fast. Now there are billions and billions, and probably in a couple of years, already now trillions of compounds that you might imagine to synthesize. And why synthesizing these very large libraries? Because people began to screen large. I'm not talking about billions, but talking about 100 millions. They noticed that, in fact, you get higher hit rates, which means that the compounds which, in fact, are selected according to what you're looking for, you get more of these compounds. Usually, if you screen millions of compounds, you will get hit rates. It depends, of course, on the complexity of the problem, but you will get hit rates about maybe 1%, 5%. When you screen very large, you go up to 20, 30%, even more. And then you get more potent hits. When you screen millions of compounds, usually you get micromolar compounds. When you begin to screen 100 millions, billions of compounds, several examples where you get 2-digit or even the 3-digit nanomolar. So you get better starting points. And then you get ligands with very, very different properties. For example, ligands which are binding intracellular signaling to a particular pathway. And this is very, it was very counterintuitive with respect to what was believed in the early 90s with the emergence of combinatorial chemistry. There are people saying that finding a drug or finding a hit compound is like finding a needle in a haystack. It's not by increasing the size of the haystack that you will find more needles. And in fact, what we observe now is exactly the opposite. The bigger the haystack, the more we find needles, and the better these needles are. Doesn't mean that they are ready to use compounds, they are just hits, but you get better starting points. I needed to, again, Marcus explained very well how these combinatorial spaces are organized, depending on building blocks. Chemical reagents that you use to build these compounds and organic chemistry reactions that you trust, that are very robust, that you can parallelize. And you just have to design a matrix, which building blocks is compatible with whichever building block to form a specific reaction. Then you can generate billions of compounds, and there is now an offer. If you go to a Biosolvity website, you will find you can access to these ultra-large libraries. Matthias Rarey introduced you with the way you can mine these ultra-large libraries in a ligand-based manner, how to find ligands which are similar, whether in 2D with respect to maximum common substructure, or even 3D similarity. So Matthias developed a lot of great tools to mine these ultra-large spaces. I'm going to address the topic of considering target 3-dimensional constraints, which of course makes the problem a little bit more complex because you have to deal with the 3-dimensionality of the ligand, the 3-dimensionality of the protein, the flexibility of the protein. And in fact, there are 3 ways to address this problem. Now I have to pray that the symbols are going to appear. Great. So either you go for, this is the American way, brute force docking, and then you screen millions, billions. Of course, you need huge hardware infrastructure to do that. But I mean, this will soon reach its limit because with the next generation of ultra-large libraries, trillion size, you cannot apply docking anymore. Even if you have the biggest supercomputer, you can't even store the data. The second possibility is that you just focus on subspaces of these ultra-large libraries that you have to select, and you just select the subspaces that you might think of interest. So you can generate focus libraries around particular scaffolds, around particular building blocks, and then design the best focus libraries around these scaffolds, dock individually each of the library. Or you can start from co-crystallized fragment, for example, and grow the fragment with specific reactions. But you need, of course, these syntones of fragments to start the work. And the last way is what Professor Shakasov told a couple of days ago, is to learn a machine learning model to predict the docking outcome. So that you learn, basically you learn a machine learning model to predict a docking score. I mean, to be honest, I'm not a big fan of this approach because I've been using docking for 30 years and I still do not know what the docking score is. Usually I don't even look at them. So, and you can apply to maybe now millions or 100 millions or maybe a few billions, but with trillions of compounds, you still need to train a machine learning model of, let's say, 1 per mil of your samples, which means that you have to train a machine learning model on billions of compounds. And this, to my eyes, is not feasible. So we have to think something about something new. And the only way to solve, try to solve the problem is to stick as close as possible to the way these libraries are built, which means we have chemical reagents and we have reactions, and is to address the combinatorial nature of these chemical spaces. So I'm going to briefly present two methods that we have developed. One is a fragment-based approach. The other one is a chemical reagent-based approach. And the idea was that the approach should be usable with reasonably limited computer resources and should be applicable to, let's say, we fix, but it's just to give an idea, at least 1 trillion compounds. So the first one is a fragment-based approach. And we designed an approach to generate new compounds from seed fragments, which are found from ligands, which have already been co-crystallized with a protein in the protein data bank. So what we're doing is that we're going to split the PDB ligand around specific bonds and generate fragments. And we're going to keep track not only of the fragment, but of the protein microenvironment as a form of cloud of points. It's like an image, but it's a 3D image. We are not using pixels, but voxels. And we keep track of 8 properties, which are basically pharmacophoric properties. Then you have a cavity you want to fill. So we don't need any ligand information at all. You just rely on the properties of the cavity you want to fill with a ligand. So you describe the cavity the same way that you describe these protein microenvironments. We have designed a tool to do that. It's called Volsite. It's a cloud of points with pharmacophoric properties. And the first step is we are going to try to find local similarities between the pocket you want to feed and these sub-pockets, these small pieces of protein pocket, for which we know we have precise X-ray data. And to do that, we use, in fact, an algorithm which is called a point cloud registration, which is used in robotics in image processing to align 2 images. The only way that we just apply it to 3D representations and not 2D representations, but it's exactly the same way. And it's very, very well suited to find local similarities between objects of different sizes. In that case here, we have sub-pockets of very, very small sizes, and we have to compare these small size sub-pockets to larger, bigger cavities. And then we try to align, to find the best possible alignment of these 2 respective cloud of points. Once we have found the best alignment, we have aligned the subpockets here. We have two subpockets aligned to the Curie cavity. We unmask the fragment, which was originally here. The fragment has just been hidden. It's not taken into account. We reveal again, we unmask the fragment, which is directly put in the 3D coordinate space of the target protein, just by applying the same rotation translation matrix that the one that is used to align a subpocket to a pocket. And then we use a degenerative linker to link these two fragments. So we started to apply it to what is supposed an easy test case. It's a kinase. It's a ATP binding site, so it's a well-behaved protein. And if the approach doesn't work on this easy test case, you can forget about it. So we use this test case because we are interested in this protein. CDK8 is a kinase. So this is the cavity here. These are the points. They are not colored, but these are the cavity points. And we run the workflow in two iterations. First iteration, we just connect two fragments together with a linker. We synthesize 37 compounds, and we tested these 37 compounds, and we already had 6 or 7 hits. And then we pick the best of the first round hit, and make a second iteration to add a third fragment to the first two by adding a second linker. And we just synthesize 6 compounds, and we generate within 43 compounds, 2 cycles, a single-digit nanomolar ligand of this kinase, which means that technically the program works very well. So then we apply to the much more difficult challenge. It's the Cache-1 challenge that Professor Chakrashov talked about. It's an open science challenge where you are supposed to find ligands for a new target for which anything is known. There is no known ligand. This one is really difficult because it's a domain of the kinase which for which you have no real binding site. It's a sort of cylindric pore, so not really druggable. Very, very, very difficult. So we apply the tool. We generate or enamine synthesize for us 94 compounds that were tested by surface plasmon resonance, and we got 4 hits, 2-digit micromolars. We were also among the winners of this competition, and in fact, whatever the method was used, no method was able to find even single-digit micromolar inhibitor. All compounds were pretty much of weaker potency. The advantage is, of course, this approach is very fast with respect to billions of docking, or even deep docking is very fast. The problem is that since it is a generative algorithm, you have to generate linkers. And when you use a generative algorithm, you're never sure that the compound you have generated is synthesizable, or even whether you are going to find close analogs, even in huge chemical spaces. And in fact, this is the hit that we got, that the program delivered. So the 2 fragments are here in green and in blue, and this is a linker which was added by the degenerative linker. And this is none of these compounds were available in real space. And we found the closest compound that we could find. So you see that sometimes the linker is not the same. Sometimes you have an extra bromine atom here, or you have an amide in the design compound. There's an amide substituent. You just have to afford the compound and to purchase the compound with the bromine. So it's not exactly the same compound as the one you have designed, but you have to live with this issue because this is a problem with any generative method. So they said, okay, can we go for even simpler and enhance the likelihood that the compounds that we design are really synthesizable? To do that, you have to go for reaction-driven approaches, where you rely on organic chemistry reactions that you master very well, and for it, the likelihood that you will generate the compounds is much higher. And we said, can we stick even closer to what bench chemists are doing? Because usually, there is I don't know whether you know the Occam-Razor principle. To the many solutions to a complex problem, the easiest one is usually the best. So let's do something ultra simple. So we start from the chemical reagents, exactly the building blocks that enamine is selling. So you have acid chloride, you have boronic acids, everything. We keep these chemical reagents totally unmodified. We don't modify them. Then we design a set of organic chemistry reactions. In that case, we are going to encode these reactions in three-dimensional space, directly in 3D. And then we're going to dock each of these fragments. You just have to dock the fragments once. So docking 135,000 fragments is done overnight, is very easy. And we look how these chemical building blocks are located with respect to each other. For example, this one, the second one, it's at the rim of the cavity. It's very far away from the first one. No way to link the second building block to the first one. Here, these two building blocks are closely related to each other. They are close in proximity, but there is no organic chemistry able to link them the way they're oriented. So we skip again this solution. Then we have the third one, which is also well oriented. It interacts with the protein. It's not very far away from the first one. And there is a known organic chemistry reaction that is able to couple these two building blocks. It's called the Borval-Hardwig reaction. And if these reagents follow the rules, topological rules, and cross-chemical compatibility, then we make the necessary, either we make the necessary bonds, or we can even make cycles. We can cyclize the building blocks, and we keep this solution. If you do that with 41 or 2-step reactions, you really end up with, in that case, almost 7 billion compounds, which are potentially synthesizable at maximum 2 steps. The problem is that the method entirely relies on the capacity of a docking algorithm to properly locate the chemical reagents, because these be chemical reactive groups which might prevent the building block to be properly anchored in the binding site for proper coupling. And to be honest, I was relatively very suspicious that can we really dock building blocks? I mean, fully or unmodified building blocks. So we generate a data set of reagents that we use by just splitting known PDB ligands which are known, for which the bond conformation is known. We split the PDB ligands. We recreate the building blocks which are required to make the bond or the cycle that we have opened, and we redock these building blocks. And in fact, whatever the docking tool, whatever the scoring function, usually in about 60 to 70%, 80% of the cases, you got a solution which is relatively acceptable, which means that the building block has been located very, very closely to the way the corresponding fragment is located in the fully enumerated compound. What is very interesting is that the accuracy of the chemical reagent docking is independent on the chemical function. So you can dock anything, carboxylic acid, even boronic acids, they dock very well. You just have the problem with the Bock protecting group, which is not easy to handle. And it's also independent on the protein to which you want to dock your reagents. We have tried GPCRs, ion channels, nuclear hormone receptors, proteases, phosphatase. Roughly, you have about 70% chances of one of your docking posts of your reagent is going to be correct. So this is a chemical space, and we carefully selected the reagents. So we started with famous Artenfeller rules, but we pay a lot of attention to the way we select the reagents which are allowed to cross-react. For example, let's take the simplest one, which is an amide bond formation. We are only going to select monoamines, which means monofunctional groups. The carboxylic acid are going to be monocarboxylic acid to just prevent selectivity issues. And we did for all these reactions. And in addition, we define what we call poisoning functions, which are side functions, which are probably going to either prevent the reaction to occur or decrease the yields, which means basically we remove all electrophilic substituents from nucleophilic reactants, and we remove nucleophilic substituents from electrophilic reactants. So that we carefully select the reagents, and then you end up with huge numbers, billions of potentially available compounds. So every reagent, every chemical reagent is annotated, by the reaction it may be part of, and its reactant role, and which reactive atoms are used to make the corresponding reaction. So it's very easy when you have these annotations to see, to recreate the bonds, the necessary bonds, or the necessary, or edit the bond types you have to make, because every atom is tagged for its functional reactivity. And when we look at how many reactions a pair of reactants can make, in fact, in 80% of the case, a pair of reactants can only make a single reaction. So the likelihood that the reaction is going to work is relatively high. You have 20% of case, or even less, 70% of case, where you might have two reactions which might occur with the same pair of reactants. So we have a cookbook, because we have to recreate this, we generate the compounds directly in the binding site of the protein in 3D. And we're not using smarts or smears. You directly do that in 3D. So we have a cooking recipe for every reaction. We have a cooking recipe with what to do, the checks, the distance, the angles, the torsions to verify. So in that case of oxadiazole, so these are, for example, two properly oriented building blocks. You have on the one hand side, you have a nitrile, here you have a carboxylic acid. Then you just make a very rough oxadiazole ring formation. And then we minimize the ligand in the protein binding site using freeform from OpenEye to just relieve the constraints or the strains which have been induced by the fact that, of course, the two building blocks are not ideally oriented with respect to each other. So let's start with a test case. Of course, we benchmark most of the reactions with known test case to be sure that we can recreate the good ligand. And then I went back to a very old friend of mine, the dopamine D3 receptor, which is a receptor on which I did my PhD in the late or mid 80s, so long time ago. We are looking for ligands for this receptor. In between, there has been an extra structure of the GPCRs with an antagonist of this receptor, which is this ethiclopride. And I was interested, in fact, by the amide bond here, because the amide bond is very easy to do. It's the largest subspace of the 7 billion. It's almost 2 billion compounds. So they say, okay, let's dock carboxylic acid and primary and secondary amines to the dopamine D3 receptor. The chemical space that we address is 670 million compounds. We generate 20 poses for every binding block. And we ask two questions. Could we find back ethiclopride, the ground truth ligand? And can we design also chemically novel compounds with this method? So this is a workflow. In fact, we are going to use a series of filters of increasing complexity, addressing, in fact, always lower and lower number of potential solutions. Of course, we have a lot of solutions at the very beginning. So just looking at whether we can recombine the two building blocks, you still have to live with 665 million solutions. You might have multiple solutions for the same ligand, because in the theory, you can have 400 solutions, which are equally good for a single ligand. And then we ask, okay, these solutions need to generate a ligand that interacts very similarly to the ground truth ligand. This to be on the safe side, not to be sure that you are going to generate a ligand at the rim of the cavity, and it's not centered the way you want to see it. So in that case, we compute the interaction fingerprint similarity to the ground truth ligand. In that case, we require that at least 60% of the interactions are conserved, and at least 50% of the polar interactions, salt bridges, H bonds, are observed. So this decreases the numbers. And so in that case, we generate these solutions in silico. Then we refine the solutions with Shipkey from OpenEye. And we said, okay, we're going to keep the solution only if the energy refinement has not modified the pose. Because sometimes, especially when you are making ring closure, then upon refinement, you completely change the orientation of the ligand. In that case, we say, okay, the refined structure shouldn't deviate more than 1 angstrom from the non-refined structure. And again, then you have decreased the number of solutions. But you still have 15 million solutions, too much. And then we said, since, I mean, the student who did this work, Francois, was the guy for which you awarded the best poster yesterday. He said, well, there are a lot of solutions. There are so many solutions that I have to filter out very, very tightly. And he said, okay, I'm going to only keep compounds which have exactly the same polar interaction fingerprint that the ground truth ligand. So they make exactly the same H bonds, exactly the same soul bridge with the same amino acids. And we asked for that overall 70% of all interactions are conserved. We have still 2,000 solutions. So we said, okay, we're going to redock all these solutions. In that case, we redock the full compound. It said, if the normal docking find again the same solution, we are on the safe side. And then we scored using a height score from Bayesian IT. And we had so many hits that we had to say, we're going to be very, very restrictive. Let's see, okay, we're going to only select compounds for which the predicted affinity, whatever it means, is below 500 nanomolar. Still, we have 463 compounds. The problem is that when you screen billions of compounds, you have millions of hits. Huh? When you screen trillions of compounds, you have billions of hits. And then you have to mine them. So, I mean, these compounds seem very ideal. I'm going to tell you the true story and not the way, not the story that we have published, because usually what you publish is not the real story. We asked NME to synthesize a few of these compounds. They synthesized 18 compounds, and we tested, I mean, we have these compounds tested by urethins for binding to D3 receptor at 10 micromolar in duplicates. No hit at all. The compound seems to be ideal. They are perfectly matching the way the ground truth ligand interact. They make the same interaction with the same amino acids. What's wrong here? This is the problem with screening ultra-large chemical spaces. Because you have so many hits that you have to find the proper way to mine these hits. And in fact, if you look at the size of your library, the number of hits that you will get in red, and the number of hits which are going to be validated in blue. We focus on the best scores. Whatever you select, we select based on interaction fingerprint similarity, but you can select your compound based on docking score, and you concentrate, of course, on the best scores always, which means this part here. So you see that the bigger your library, 1 million, 100 million, 10 billion, the more you will find solutions here in this part of the plot. When you look at the hit rate, I mean, the compounds which are experimentally verified, you see that, in fact, within this board, I mean, the hit rate is pretty much standard, which means that you have here 100,000 of compounds, and there is no reason to select really the best of the best one here, but you can select any of the compounds here, and the way you select this compound is very important. And of course, to select 50 compounds out of 10,000, there are infinite ways of selecting this 100 compounds. So we have to be more rigorous in the way we select our compounds. We have here presumably ideal solutions. So the similarity is 1, which means exactly the same interaction that the ground truth ligand. You see the ground truth ligand is here in orange, and these are the hits, so that overlap very well. They make the same interactions. I mean, the predicted binding affinities, the height score, I don't know whether Marcus is still here, the height scores are very, very high, so they are very, very high, and these compounds do not bind. So there's something that is going wrong with the way we select the hits. So we went back to the data. I said, okay, we're going to try to automate the way we look at poses if these poses were rare. So we look really individually, one after one in 3D, how these compounds bind to, and we're going to try to automate the human reasoning we do. Let's first eliminate the strain torsions, which means that just the dihedral angle values, which are presumed to be not compatible with statistical distribution of this corresponding angle distribution. And for that, we use a nice tool, which is a torsion analyzer from Matthias Rahay. And we don't want to see any strain torsion at all. This filters out a lot of solutions. Then we are going to look at unsatisfied polar atoms, which means polar atoms of the ligand, which do not interact with the protein at all. And of course, this is enthalpically disfavored. And we don't want to see any ionized positively or negatively ionized group unsatisfied with respect to protein interaction. We tolerate at the most 2 unsatisfied H bond donors and at the most 3 unsatisfied H bond acceptors. This removes a lot of solution. And then we are going to look at the global and the local strain energy, which means the energy you need to bring your pose to the closest minimum in water or to the absolute global minimum in water. And to do that, we use free forms from OpenEye. So these thresholds were defined, in fact, by looking at PDB structures. And if we, in fact, we use these thresholds, we would only discard only 25% of all PDB ligand complexes in the PDB. So relatively robust procedure. So we went back to the raw data and applied these filters here. And of course, got different hit lists. In that case, we only got 315 compounds, which are different from, some were in common, but some were different from the first hit list. And the ground truth ligand was ranked 33. We said, okay, this sounds good because we're able to recover the ground truth ligand for the good reason. And we went for a second batch of screen. So we selected only 15 compounds, again, the same procedure. And then we got of 15 hits, 10, which were binding to the receptor at least more than 50% inhibition at 10 micromolar. And out of these 15 hits, 6 hits were sub-micromolar ligands, and especially the red ones were totally new scaffolds, I mean, patentable compounds, which was quite surprising because there are hundreds, thousands of dopamine receptor ligands which have been synthesized, and still you can find compounds that you can you might patent with easy organic chemistry. So the hit rate at 10 micromolar is 67%. The hit rate at 500 nanomolar is 20%. So this agrees with the tendencies we observe when we screen ultra-large library with full atomic docking. The bigger, the higher the hit rate, and the better the hits. The full procedure is relatively fast. The space docking is relatively fast. It's about 1 week. The hit selection, 2 days. In synthesis, in amine are really fast. 5 weeks. I think now they can synthesize within 3 weeks the compounds you order them. And in vitro assays 2 weeks. So we start from scratch. After 2 months, you have your compounds. Then you say, okay, we are now using this method routinely with more difficult targets. And there's the question: how far can we go? How much can we screen? Is there any limit? Is there any reasonable limit? And to increase the size of the chemical space, only ways to go for either multi-reactions, multi-step reactions, or to go for, in that case here, in 3-component reactions where you use 3 building blocks. So here the challenge is to correctly position 3 building blocks with respect to each other. So we use this example, which is the petasis reaction between an amine, an aldehyde or a ketone, and a boronic acid or boronic ester to form this tetrasubstituted amine. In fact, we haven't chosen this reaction randomly because, in fact, there is one ligand in the PDB, this compound, which is an inhibitor of this P300 acetyltransferase, which is a very, very important, it's an epigenetic rider. It's a protein which acetylates lysine residues on histone. So very, very important for DNA editing. And very important target in biology. And the ground truth ligand is synthesizable by a 1-step petasis reaction. So we start again from the stock of building blocks of enamine. We select the reactants based on smart strings. We convert the reactants from 2D to 3D, of course, because we have to dock 3D. And in that case, the chemical space is 360 billion compounds. We say, oh, this begins to be huge. Let's see where we can do it and what is needed to screen 360 billion compounds. So I'm going to pass the filters. It's the same procedure. We dock, we look at the compatibility of the building blocks. In that case, you have a triad of building blocks, not a pair of building blocks. We refine, we do the in silico synthesis, we refine the structure, we filter out by quality check, we look at the availability in real space. We have 196 compounds. The compounds are going to be tested next week. Sorry, I cannot disclose the results. But whatever, I mean, what was important is which computer resource do you need to do that? In fact, it's not that much, but it's something that you usually see in the papers. We look at every step of the workflow, what is required in terms of CPU hours, in terms of disk storage, in terms of memory usage. And you see that, in fact, the docking doesn't need that much. It's very, very light. And hopefully, of course, the method doesn't scale with the number of reagents. It scales with the number of potential recombinations of your building blocks. In that case, you need a few, about, in that case, 100,000 hours CPUs, which is relatively a big number, but not that big. If you have, let's say, 500 cores, you can do it. Energy minimization, EFP similarity, redocking of the ligand, quality check is very expensive because freeform is very expensive. You generate conformers in water, so this takes time. So if this work was done over 3 weeks using 3,000 cores, we have done this screening using brute force docking, using the same amount of cores. It would have taken 20 years. So the acceleration is still very, very, very high. But when we look at the workflow is very inefficient because, in fact, you spend most of your time in rejecting solutions. These are the solutions which have been accepted by the space docking procedure. It's 0.00005% of the solution. So we spend most of the time in discarding solutions. So what's the question? Can we predict in advance the reagents and the reactions that must be used to generate target-specific ligands? And then we try to generate a model would predict which reactions and which building blocks to use in advance, and only apply the space docking procedure to these building blocks. So we use a native loading protocol. We start from the chemical reagent and we select a very small subset, 0.1% or 0.2%, which is a training batch you are going to use to train your model. And we space dock these small samples of building blocks. So the building blocks are spread everywhere. You see that these two building blocks are not connectable. This pair is probably connectable because they are close to each other, and they might be maybe linked by one of the 40 organic chemistry reactions that we handle. So that we are going to fingerprint the pair of reactants and the reaction that is made in a fixed length fingerprint. And we label this fingerprint with respect to the success that the space docking procedure has to recombine or not these building blocks. So that you can train a binary classifier to generate a pair of reactants that are supposed to be suited for your target of interest. And then you can select an additional subset by different methods I'm going to present you to increase the size of your training batch, dock again, reselect again, and then you can repeat this active learning cycle several times so that every time the model is going to learn better and better which are the reactants and which are the better reactions to use with these reactants to generate target-specific ligands. And we applied the procedure to 2 test sets for which we did the full job. So the first one is the dopamine case I told you about with 370 million of compounds, which means 670 million of reactants and a single reaction. And then a more complicated case, test this one, for which we have a chemical space of 7 billion compounds and 11 reactions to define this chemical space. The hydrates are very different. So this is a true space dock hydrate. The percentage of building blocks that we're able to recombine to enumerate a full ligand that interacts the same way as a ground truth ligand. In that case, the hydrate is 2%. In this case, it's 0.5%. So, and these are the active learning parameters. So we vary the initial batch size in terms of pairs, the size of the exploration batch that you use to extend your initial training set, the acquisition method. This is the way you select new samples. So either you go for what we call a greedy selection. You take the best probabilities, the highest probability. Or you can this is a pure exploitation strategy, or you can use an exploration strategy, which you select the pairs for which the prediction is the most uncertain. So in that case, you're going to explore a lot of solutions. Or you can have a mix, which is called template sampling approach, which is a mix of exploration and exploitation. And we derive different models. Simple logistic regression. My advice is always start with simple. Then more complicated, random forest, and then multi-layer perceptron. And the performance was examined by looking at the space dock recall, how many of the hits we recover by sampling the chemical space. And these are the results. And in fact, we have increasing definition or stricter and stricter definition of hits. This is the interaction fingerprint similarity that we use to define a hit. So in that case, we ask the pairs of reactants should be recombinable, but the ligand should exhibit at least 60% of common interactions with the ground truth ligand. 70%, 80%, 90%. And you see that by just exploring 5% of the full chemical space, you recover, depending on the way you define it, 75, 80, up to, in that case, if you are very, very, very strict, in that case, 98% of your hits in advance. I'm almost finished. And for the second one, it's slightly lower because the number of hits is really low. Huh? In that case, there are only 42 hits out of 7 billion, but we could find 26 of these hits in advance. So you can use it to, in fact, to restrict the chemical space. My take-home message, the ultra-large chemical space are really modifying the way we address virtual screening. I've been doing virtual screening for almost 30 years. I would have never imagined that we screen 100 billions of compounds now, which is really affordable. The second message is chemically novel hits can be synthesized by simple organic chemistry. You don't need to go for very fancy chemistry to generate hits. I'm not only talking about hits, just generate hits. I was educated as a medical chemist. I'm still a medical chemist. I'm asked the question, but do we really need in-house medicinal chemists to synthesize these virtual hits? Because enamine is so good, so cheap, that there's no way we can compete with them. So that I've totally externalized the virtual screening hit synthesis to enamine, because no way that you are going to spend a postdoc to generate compounds at a very low throughput when enamine can generate hundreds of compounds within a couple of weeks. Do we still need in-house medicinal chemists? Yes, of course, we still need medicinal chemists because, and this is going to be my last message. A hit is not a lead. Lead compound has more properties. A lead is not the clinical candidate, different properties which are needed, and the clinical candidate is still not a drug. To address these issues, you need to go and to predict much more difficult properties, especially in vivo properties, for which we do not really have nice solutions. Okay, this is it. I want to just thank the people in my lab who did the work. Mostly the work was done by François, Enamine for the nice collaboration, the screening platform in Strasbourg, and the IGC in Toronto for the experimental validation. Thank you very much.
Great. So one short question. Here's a short question.
Reaction you had, could you follow up with enamine if they really did amide formation reactions? Do you know?
No, because well, when this is amide bond formation, probably they're going to use it because it's the simplest one. But sometimes it happens that, for example, the petasis reaction is not part of the enamine catalog.
Ah, okay.
They don't do it.
Okay.
But this doesn't mean that you cannot do this compound with other routes. With reductive amination, you can do the same compounds.
Yeah.
So they don't tell you which routes they are using. I mean, you can ask, then you have to pay to get the experimental detail. Okay. But usually they use the same route because we start from the reagent that enamine used to define the real space, and we generate their reactions. So probably they are going to use the same reactions, but we are not entirely sure.
Okay.
Okay. Thank you very much.